Welcome to the Cannabis Cultivation and Science Podcast. I'm your host, Tad Hussey of KISS Organics. This is the podcast where we discuss the cutting edge of growing from a science-based perspective and draw on top experts from around the industry to share their wisdom and knowledge. My guests this week are Victoria Holden and Charles Smith, with a short appearance by Ben Higgins. Victoria and Charles are the co-founders of Imio, and in this podcast, we discuss their products and research. They currently have brought two products to market, an organic, microbial-based rooting powder and gel, and a microbial-based solution for breaking down biomass faster, which has broad applications. This podcast is over a year in the making, as we wanted to run trials at Gold Leaf Gardens with Dr. Ben Higgins to see how the organic cloning product worked and get some independent data on our end. There are so many microbial products on the market these days. It's very difficult to separate out the marketing from research and determine what's worth using in your garden. And we wanted to make sure that all the products we offer at KISS Organics or talk about on the podcast have legitimate research and value. Victoria has a lifelong interest in studying the interactions between microbes and their surrounding environment. She earned her Ph.D. in Microbiology and Immunology from the University of Michigan before moving to Dartmouth College to further her studies with postdoctoral research. As Chief Science Officer of Imio, Victoria has led the development and productions of multiple microbial products that replace harmful chemicals and enable healthier, more productive cultivation of plants. Charles started Imio to dedicate his career to helping people, plants, and our planet. After graduating from Dartmouth College, he began his career at Morgan Stanley, where he focused on the technology sector. He moved on from finance to join a New York-based software startup where he gained valuable insight into scaling companies and teams. As CEO of Imio, Charles manages business development and operations as the company works to unlock plant productivity and restore our planet through the power of nature. Now on to the show. Hi, Charles. Hi, Victoria. Thanks for coming on the show today.
Thank you for having us.
Yeah, thanks for having us, Tad. We're thrilled to be here. Really enjoyed the podcast. You share a ton of great knowledge here, and we're very happy to be part of the conversation.
Yeah, so you are co-founders of Imio. I met you guys through another connection, learning about some of the products that you have, and I came into this sort of a little bit skeptical, but through some trials that we ran, which we'll talk about, and some of the other trials, I'm really excited to talk about what you guys have going on in your company. So let's just start off with a little bit of an introduction. Charles, I know you guys are co-founders. Can you tell me a little bit about your role and who you are?
Absolutely. And I just want to say briefly, Tad, totally appreciate the skepticism. It's really good to kind of put products through their paces. And, we appreciate kind of the data-driven approach that you took to getting to know our technology. Mm-hmm. In terms of kind of backstory, I'm the co-founder and CEO of Imio. So I lead sort of our business development and operations. Victoria is our chief science officer. So she's the real technical brains behind actually making these products a reality. And the backstory on Imio, started with really wanting to dedicate our careers into doing good things for the planet, and recognizing the huge opportunity that natural microorganisms offer in terms of helping plants grow more effectively and also recapturing valuable nutrients from things that are otherwise treated as waste.
Wonderful. And Victoria, can you tell me a little bit about yourself as well?
Yeah, absolutely. So I'm a microbiologist. I earned my PhD in microbiology from the University of Michigan. I've done a lot of research studying how microbes interact with their environment. For a part of my career that was within the human body. And then I made this transition into wanting to understand more about how microbes interact in our natural environment and in the world. So I was really fortunate to meet Charles at a good time in my career when I was ready to transition into doing something that could really help the earth in a more immediate timeline than compared to, say, academic research. And very happy to be helping to lead the scientific development and production of Imio with Charles.
Awesome. So before we dive into the products and the microbial process itself, can you tell me a little bit more, I guess a little more big picture about how you founded the company and yeah, what that all looked like?
Yeah, absolutely. I'll kick us off there. So, as I sort of alluded to in my introduction, um, it started with recognizing what we see as a huge environmental problem that also presented a really major opportunity. Um, Specifically, what we started the company focused on is the amount of organic matter, food and plant matter that is treated as waste, and the environmental repercussions of that. As you probably know, Tad, about a third of food produced globally is wasted every year. That has huge repercussions in terms of greenhouse gas emissions, particularly with the production of methane, which is an especially potent greenhouse gas. Um, and as I was kind of digging into that problem, wanted to come up with ways that sort of the value in that wasted plant matter could be unlocked. You may notice that I try very deliberately not to say plant waste or food waste, because we actually like to say there's no such thing as plant waste. A lot of plant matter is wasted, but there is a lot of value potential that's left in that, in the stalks and the leaves and everything that's left over, not eaten or sold or what have you. Um, and of course, as you know very well, microorganisms are the way that nature deals with that challenge, turns stalks, leaves, etc., into nutrients so that the next generation of plants can grow. And our idea was that if we can develop a combination of microbes that can do that job really quickly, consistently, efficiently, we could make it much more accessible for more people to recycle their plant matter and do really good things for our planet and for, of course, the plants that people are growing both at home and commercially. So that product became Regen, which is the first product that we started the company around. We got some funding from the National Science Foundation to develop that product. Victoria can speak a little bit more to that. But then as we kind of, we're getting out in the market with Regen and talking to a lot of really great growers of all sorts of plants, but especially cannabis, we came to understand sort of the really large world of opportunity for microbes to do really good things for people to grow plants health in a more healthy way that would be good for growers in terms of their output and much better for our planet, both in terms of how we eliminate quote unquote waste and also replace chemical inputs with naturally occurring microbes.
Yeah, and I have some questions for you next, Victoria, but I have to say the Regen product I think is really interesting. And I'm still wrapping my brain around some of the applications within cannabis. I absolutely see them within agriculture. Here in Washington State, the way we dispose of plant matter makes it a little bit problematic. But I would like to dive into that a little more with you. But what I was so excited for, and this is something I think it goes back probably almost a decade now, within the organic cannabis side, there's never been a good organic option for cloning and propagation. I did my own little trials and experimented and ended up with a huge moldy mess trying to create my own. Back in the day, you hear people using aloe vera with some success, but not being a microbiologist, I had to go like on the fertility route, trying to figure out a solution for propagation. Now, you guys came up with a microbial solution for propagation that I think is really interesting. Victoria, can you talk a little bit about that development process? Like, how did you go about once you guys decided, you wanted to create Regen or you wanted to create Root, what that looks like on your end as a scientist?
Yeah, so I'll start with Root first, just because that's one you mentioned being particularly excited about for this conversation. When we came up with the idea to create this organic, natural cloning solution, we first kind of looked at what else growers were using and what were the components of that product that either helped it work well or caused these other less exciting potential drawbacks to its use. So the main things in synthetic cloning solutions that are good for the plants are indole 3 acetic acid and gibberellic acid, which are two hormones that help develop roots. And these are both hormones that microbes can produce themselves. And so we simply went to published research, read a lot of papers, and tried to find microbes that are naturally occurring that produce them on their own at concentrations that could be useful to plants. We tested a few different combinations of these microbes. We tested the microbes individually and then started applying them to plants and seeing which combinations could help the plant's root in a way that didn't involve these synthetic chemicals that can be really harmful to people, plants, and planet, frankly. So it really started researching what other people have already done, bringing them together and moving that knowledge forward to create the root product.
So once you identified, a particular strain of bacteria or, a fungi, I don't know a ton about the product, to be fair. I've talked about it a little bit with Charles, but once you identified this, these, how did you go about collecting your initial specimens and then determining what sort of synergistic effects you might be looking for getting from the microbes themselves?
Yeah, that's a great question. The way that we proceeded to source these microbes was by acquiring them from strain collections. So similar to seed banks, there are microbial banks where you can purchase them and then work with them in your own lab. So that's predominantly the way that we have sourced the microbes en route, though we have done some work with isolating microbes from the natural environment around us. But for the time being, root contains microbes that we've acquired from these microbe banks.
Piggybacking on that a little bit, one of the reasons that we take the approach that Victoria described in kind of identifying what we describe as candidate microbes and then sort of vetting them first through research, then of course with our own experimentation individually and in a consortia. It's really important to us that we know exactly what is in root and regen and all of our inoculants, because we want to make sure that, first of all, we can, ensure that we're avoiding spreading any sort of potential pathogens, anything that could, replace what we see as a big chemical problem with a biological problem. Our sort of vetting process involves immediately eliminating any, candidate microbes that could become problematic for people or for animals, or of course for the environment if they were to be ingested or kind of be in a place where we wouldn't expect them to be. So again, that was sort of part of our rationale. Let's make sure we understand exactly what's going on here. And the other, sort of ongoing benefit to that from a practical standpoint is it's really helpful for production consistency. So again, knowing sort of inside and out every individual bacteria and fungi that is in each of our products allows us to produce them very consistently, first as an individual microbe and then ultimately combined in a particular ratio that we found to be most effective.
So most of the products for propagation, if we're talking about root here first, are they're not microbial based. Is that a fair statement based on your? I mean, you guys are more familiar in this area than me. The ones that I see are, as far as I know, salicylic acid or IBA products. Mm-hmm. And the goal was with this microbial product to get comparable or better results, right? I think that was what we talked about. So, how, knowing that this approach differs, what sort of results are you guys getting with root using microbes instead of direct chemicals?
Yeah, we get great results. We have, we're always aiming to be the best product out there. And for a lot of strains and a lot of different growth medias, we do have the fastest and most robust rooting. For some strains of cannabis, we're on par with those synthetic competitors, which is actually a result that our trial with you showed. One strain root did better than the competitor, and one strain did exactly the same. And we feel great about that for a handful of reasons. One is that it's a real scientific living microbial product. It's not going to be perfect all the time. It's okay that it is as good as, but often better. It's safer. It also provides better long-term results because you are giving these plants symbiotic endophytic microbes that benefit their whole lifespan and not just that initial rooting stage. So the benefits are even longer term. And so those are some of the results that we're seeing from our growers now, both better rooting, faster rooting, shorter time in veg, higher yields, more successful cloning all around. And we feel great about that.
So for a layman like myself, essentially, as I understand it, then would be I'm getting the same chemicals as I would in Clonex or Dip and Grow or these other commercially available products, but I'm getting them from this microbial interaction with the soil and water and plant that then allows for potentially at least comparable, but potentially better results down the road. So let's say I get 95% propagation success. Where you're seeing the big differentiation is as the plant starts growing its true leaves, as it gets a little bit bigger through veg, you may see some persistence with these microbes that would lead to better veg growth compared to the control of Clonex or Dip and Grow or these other commercially available products. Is that right? Or do you want to add to that or talk about some of the research that you've done?
Yeah, that's right. And happy to sort of add some of the data that we've gotten back from some of our customers and growers that we work with closely. As Victoria described, because root creates this symbiotic relationship with the plants, the benefits do really, continue as the plant keeps growing. Sort of that the difference between a chemical shock from a Clonex or a Dip and Grow that, of course, is effective in forming roots, but then kind of goes away. Whereas the root microbes, continue to live within the plant tissue, within the roots, within the substrate. And that has translated to really, exciting results in the vegetative growth stage. Some of our customers have noted, reducing veg cycles from 30 days down to 20. I think the biggest change we had was maybe like a 28 down to 14 cycle for sort of the kind of veg stage parameters that this grower was looking for. And you kind of mentioned, Tad, the interactions within the soil. Of course, root is a really exciting way to introduce microbes into the soil, but also into the plant itself, kind of taking advantage of the access that is created when you dip that cutting directly into this microbial consortia. And that's something that we've seen translate to really exciting results also in rock wool, in cocoa. Of course, we're big believers in living soil ourselves, but we've also seen kind of the benefits of using root in soilless substrates as well. And in some ways, it's kind of a unique introduction to microbes in an environment where they can thrive within the plant, even if the substrate is not kind of optimized for microbial life.
So you're seeing good results in Rockwool, for example, or Cocoa Core, where you may not have any microbial ecology. So even with the chemical mineral salt program, you can still see results, is what you're saying.
That's right.
Okay. All right. So tell me about what's in root.
So, root is comprised of um, a few different families of microbes that all provide some good benefits to the customer. The main category that provides a lot of those benefits are the indole 3 acetic acid, or IAA, and gibberellic acid producers. So these microbes make natural versions of those hormones that are often included in products like Clonex, Hormex, etc. And then another category of microbes that are included in our product are endophytes. So these are microbes that can actually enter the vascular tissue of plants and form symbiotic relationships and help the plant with things like its health and vigor. So those are two really important components of root product. And then root also contains a natural gelling agent called guar gum that's isolated from legumes. So this is the product that helps form that gel that growers really often like to dip their clones into. And finally, it contains a little bit of yeast extract and sugar, and those are just included to help the microbes have something to eat when they are initially woken up from lyophilization or the freeze drying process.
Now, speaking of microbes, one of the things, and one of the reasons for my original skepticism is just there's a lot of people there's a lot of companies and products on the market right now that are microbial based. It's one of the fastest growing segments of, I think, agriculture in general right now, which most people call biostimulants. But some of them are just sourcing bulk powders from China. They'll buy Bacillus subtilis because you can read about some of the research around that. But what are the differences with Imio and the advantages associated with culturing your own microbes and controlling that process versus what many of these companies do? They're just buying these powders and essentially mixing these microbes together.
Yeah, I think our biggest advantage with the way that we do it, which of course is we grow our microbes from a frozen 1 milliliter microbial stock into tens of liters of microbes, is that this allows us to control every step of the process. It allows us to have a lot of room for quality assurance and quality control. And it helps us make the product really consistent because we're growing each microbe individually, combining them at the ratios that we have determined to be the most beneficial to plants, and checking them along the way through all of the steps of the process. Because, as I've mentioned, these microbes are alive, and if you aren't able to provide them at the right stage of their life cycle, they could be much less consistent, much less beneficial, or oftentimes dead, unfortunately.
Yeah, that's a real concern with on-shelf products. The Oregon Department of Ag, I've had them on the podcast, and they shared some of their research that they did independently. And it may not be a reflection of the original quality of the product, but the fact it may have been stored in a warehouse, exposed to sunlight, heat, temperature extremes can definitely degrade a product. So the way you're structured right now is shipping direct from your facility to the grower itself. So if, for example, if someone ordered root through Kiss or Regen, it would ship directly from you to them. So there wouldn't be an issue of expiration dates, freshness, or quality.
Correct. And we do a lot of work to maintain and check on the quality. So As I've mentioned, we check that the microbes are still alive at various stages. We do a lot of our own experiments with cannabis clones and other plants to make sure that the product is working at different time points of its lifespan. So we feel really strongly that that's the work we have to do on our end to make sure we're providing customers with a consistent, strong product.
To your point about kind of shipping directly to growers, that was the only way that we would operate for kind of the earlier stages of the company when we were shipping out a liquid or gel version of our product directly, because that of course was a live culture. It was active, and as you described, we needed to be very conscious of time, of temperature, even thinking about things like pressure fluctuations, anything that might happen in transit that could damage the health of the microbial community. Because of course, as you both were just saying, the microbes have to be alive to form a symbiotic relationship with the plant to provide the benefits that they can offer. Victoria and our whole science team have been very hard at work on a freeze drying process called lyophilization, which Victoria can expand on, but which essentially allows us to take each individual microbe, run it through this lyophilization process that controls both temperature and pressure to remove and freeze the moisture from the microbes without exploding their cells. And so now we can send out a shelf-stable powder that is, essentially has the microbes in a dormant state until water is added and the product becomes ready to use when the grower is ready to use it. So that was, kind of an essential step for us from our perspective before we would, kind of entertain any possibilities of there being any steps between, us and the growers. As you described, we of course continue to love to work directly with growers, and working with KISS is going to be a great way to kind of bring more people into that network. But as you said, to also send them directly. But just wanted to kind of emphasize, the efforts around consistency and ensuring viability. It takes every step along our production process, and then it's kind of accumulates with this lyophilization, this freeze drying process, so that we can take out kind of the variables of could something go wrong in transit, because if it's a live liquid product, the answer to that from our perspective is absolutely yes.
Another question I had, because this is becoming a bigger issue in cannabis, is heavy metals. Can you talk about that a little bit?
Yes. So specifically, I can say that we have had both of our products tested for heavy metal concentrations. We're currently going through all the state's regulatory processes with the different departments of ag. So that is a requirement of those processes, and our products are all set on that end, which is wonderful. Around Regen, which is our recycling product, we have definitely talked to growers who have talked about the fact that cannabis can be used as a bioaccumulator plant, and that when it grows, it can absorb the heavy metals from its environment. And so it is a concern of repeated cycles of regen recycling on hemp or cannabis that has grown in heavy metal rich soils. What is the impact of that? And there's still a lot more testing to be done. But the first test that we've done, it seems to be not a problem. We haven't done repeated cycles of regen growth and then tested the heavy metals on that. So that is something that we are very interested in doing. And again, like Charles said before, small team, lots of questions.
One thing I just add to that is, the guidance that we tend to give growers if they're concerned that they're sort of, on the edge of permissible levels of heavy metal, we recommend that they do not recycle the root ball in that scenario. As I think Tad, the roots tend to be an area where heavy metals become particularly concentrated. So sort of taking that out of the recycling stream, if you will, ideally having it end up in another kind of compost process, but not immediately reintroduced back to your soil or your own grow. That's the way that we've handled that in the past. And as Victoria said, all the testing that we have done leads us to be comfortable with the composition of the product and sort of the recycling process overall. But that is kind of the guidance that we give, is think a little separately about the root ball versus the stalks and the leaves.
Yeah, I guess when I asked the question, I was thinking more in terms of the product itself, like root and regen, not so much the material you're working on. That being said, the amount that you're adding in root or even with regen is so minimal that I don't think it would be a huge contributing factor to the overall heavy metal uptake or analysis. But you have tested your products then, and they are passing in the various
Oh yeah,
state regulations. So that's good to hear.
Yeah, way below any levels that would be concerning, to regulators or growers.
Yeah, I'll have to get that from you to be able to have that published on the website too.
Yeah, sounds good.
Let's talk more about the research now. What sorts of results? And I'll get with Dr. Ben Higgins and talk about the internal trial that he ran over at Gold Leaf Gardens and his results separately, and we'll tack it onto this podcast at the end. But tell me a little bit more about the other research that you guys did. You did some work with UVM, it sounds like, and then a few different facilities. One of you wants to jump in on that?
Yes, we love collaboration. So our work with the University of Vermont was actually on primarily our regen product. So as Charles mentioned, we were awarded a National Science Foundation Small Business Innovative Research Grant in 2020. And so as part of this grant, we were developing different formulations of regen and then passing them off to UVM for them to test as a third party. So that was great. We had a very strong collaborative relationship with Dr. Deb Nair and her graduate student, Noah Olson there. That research is actually under review for publishing in a journal called Compost Science and Utilization that we hope will be accepted very shortly. So we would love for everyone to take a look at that if they're interested. And then with regards to root and regen both, we love to set up as many collaborative products or projects with growers and farmers as much as we possibly can. We think there's a huge value in this kind of third-party validation and trusting expert growers to insert our products into their processes and then give us feedback on how they work. So Charles, I don't know if you wanted to speak a little more to that.
Happy to. And I'm glad you gave a shout out specifically to Dr. Deb Nair and to Noah Olson. Noah, I know is an avid listener of the podcast. So and as you said, Victoria, UVM has been a great partner and collaborator to us, generated tremendous data on regen, quantifying not only the plant outcomes, the yield improvements that can happen with regen in recycling plant matter, but also in really quantifying the microbial actions, the ecoenzyme outputs, to help us really understand, the mechanisms of how these microbes are breaking down the lignin and the cellulose, the really tough parts of the plant matter, in order to create access to the nutrients that they contain. And we've also, had a wonderful experience collaborating with a lot of different great growers in Vermont, in California, Colorado, Arizona, and have generated tremendous data. As Victoria said, we really value, seeing, what regen and root are going to do in a live, commercial cultivation environment where there are all sorts of different variables at play, and you need to make sure that the kind of product and the the science is really strong to make sure that you get consistent results across all sorts of different grow types and conditions, which is very much what we've seen. So I'll mention a few specific results that we've gotten with root. One of our customers out in California is a grow called Floraterra. They've got a really strong cultivation team, several different retail locations as well. They shared with us that when they switched from Clonex to root, they had a really wide range of benefits associated with that transition. They had been struggling significantly with a fungal pathogen that was really giving them a lot of trouble in their cloning process. So they had a cloning success rate that was way below their targets. They had to kind of keep taking more cuttings from plants that they didn't necessarily want to kind of put into the mother plant category, and unfortunately we're caught in a really tough cycle. Since transitioning to root, they have gotten their cloning success rate way above their target rate. They're now around a 95% success rate, and mind you, they're doing at kind of peak clone times, they're doing about 20,000 cuttings a month. They are no longer struggling with the fungal pathogen. They've increased their yields. They've set room records with plants treated with roots, and they're one of the examples that I mentioned before in terms of the accelerated vegetative growth times. They had been running 28-day veg cycles, and in switching to root, they're down to 10-day cycles.
That's a massive difference in savings in electricity and other resources. That's great.
Totally. We've been really, excited to be part of that transition, and grateful to them and their meticulous, data tracking to be able to share such clear results with us. As you know, Tad, doing good science takes a lot of, focus and data. And so, as I said, we're grateful to their team for, all the tracking that they did to provide us with these really clear results.
Great. Well, I will have Ben on to talk more about the results at Gold Leaf. So, just in the interest of time, I think I covered most of the questions that I had for you guys, at least around roots. I did want to talk a little bit about Regen. It's not a product that we tested internally for some of the reasons that we talked about. I guess you and I had dinner together, and we talked about some of the applications outside of the cannabis industry. But why don't we talk about some of the potential uses for that product and how people are using it, and what the results might be. And I can ask questions around that, because I have a few more about Regen itself. But yeah, why don't you just go ahead and share some of that?
For sure. Victoria, you want me to get us started there, and you can kind of jump in?
I guess I'll explain the science behind it a little bit, and then maybe you can talk about the applications of it.
Perfect.
The idea behind Regen predominantly centered around the fact that hemp and cannabis in particular have a really high concentration of this difficult to degrade molecule called lignin. So for fellow science nerds out there, lignin is very aromatic rich. So it forms these carbon rings that take a lot of energy to break down. But behind and underneath all of that lignin are cellulose, hemicellulose, and all of the nutrients that help the plant exist and continue to grow. And so when you think about breaking down cannabis to release those nutrients, you first need to break through the lignin. And so that is the main molecule that we were thinking about breaking down when we were looking into candidate microbes to bring together into our Regen product. There are a few different enzymes that can be used to break lignin down. We found quite a few microbes, bacteria and fungi that are able to produce those enzymes. We tested the individual microbes' ability to produce those enzymes, the consortium, the various formulations that we tested and their ability to produce those enzymes. And based on all of our different combinations testings, I think we ultimately tested like over a dozen different formulations of regen before we found the one that worked the best and that we were most excited about. Then we were able to hand that product off to UVM for them to test it in some more eco-enzyme assay types of ways. So, and then recycling the plant matter. So we love that product. It's very conceptually exciting to the both of us. It's, of course, in a lot of ways what got Imio started due to that NSF grant. So I'm excited for Charles to talk a little bit more about the applications of it.
Yeah, and just to echo Victoria, it is kind of the product that we see as kind of the biggest idea, the biggest potential impact that we can have, around kind of agriculture and the environment. Where, of course, as an environmentally focused company, we're very excited anytime we can eliminate a harmful chemical, and root is certainly a product that does that and leads to better plant outcomes while, taking away what can often be a dangerous chemical input. But regen, as you described, Tad, can be used in an agricultural setting, can also be used in commercial cannabis cultivation or home gardens as well. Anywhere you've got plant residuals and kind of a demand for nutrients. We've had we've done a lot of tests comparing side by sides with various fertilizers, various composts, with and without regen, and we've seen yield improvements for plants treated with regen in the range of 16 to 35% on average, including some tests that we've done with some really strong living soil grows around the country. To your earlier point, Tad, there are sort of some regulatory dynamics that vary from state to state. Some states may be more or less open to the idea of on-site recycling of stalks and leaves. But in the scenario where the kind of regulations are open to on-site recycling, we have both indoor and outdoor growers who will collect their stalks and leaves, put them in super sacks or into piles, apply regen to the top, mix that up a little bit. More mixing can lead to more complete breakdown. It's not required, but it can make things go a little faster. Those lignin degrading microbes tend to like oxygen. But that recycled organic matter can then be applied back into the soil, mixed in as an amendment or used as a top dressing. We've also found that, particularly in the top dressing application, the water consumption requirements go down. Generally, as you know, mulch has a lot of strong benefits for that, and the regen recycled biomass is an especially nutrient and microbial rich sort of mulch or amendment. So those are the different applications that we see there. And then we've also done a lot of work on corn farms and in forage crops, spraying regen into the field, allowing those microbes to do their work to break down the residual plant matter into nutrients for the next cycle. And that can also be done, at small scales on leaves in backyards. Again, we're really big on this idea that there's no such thing as plant waste, and regen we see as a way for people to kind of recognize that and make it easy to get all the benefits back from something that in some instances they may be paying to get rid of.
Yeah, I love the idea of using it for like silage or plant matter that's left out in the fields, agricultural, to break it down faster for the next cycle, or maybe breaking down a cover crop after cutting or things like that. I do struggle with, and talk to me about this. recycling plant matter in and out of a facility raises the risks of pests and pathogens in controlled environment agriculture, which is what a lot of our growers are doing. Now, granted, with living soil and such, we do give up a little bit of control already by recognizing that there's going to be microbes in our facility. There's going to be beneficial insects and things like that, but we're still trying to avoid pests and pathogens. How does regen impact those risks, or how do you see it playing a role in a situation like
For
that?
sure, it's a great question, and before I kind of answer it, I'll just say I think, Tad, in the way that we did with Root, we're going to have to prove it to you with Regen. And
Okay.
again, very very much appreciate that. Um, to your question about pest and pathogen risk, we of course recognize that that's a huge consideration for commercial growers, particularly, as you said, operating in an indoor controlled environment. Um, we were able to generate really strong data with UVM, um, both into kind of the mechanisms of what's going on with the microbes, and also how that translates to interactions with different pathogens. So I'll kind of point to the research paper that Victoria mentioned, which will be published soon. UVM performed side by side. They performed what's referred to as a plate competition assay, in which a pathogen is grown on a plate. And then you kind of do something to the plate to see whether the growth of that pathogen is impacted. In the case of this experiment with UVM, they used the fungal pathogen Rhizoctonia solani, which is a particularly strong fungal pathogen that also is used often in academic settings because, one, you can grow it on a plate, and also because it's sort of representative of a bunch of different types of pathogens. And in those experimental conditions, they found that putting recycled biomass that had been treated with regen onto that plate with the fungal pathogen prevented that fungal pathogen from growing. There was full suppression of the pathogenic growth in that context. I'd also mention, to your point about pests, of course, we think about insects that we don't want running around or flying, crawling, et cetera, around the grow rooms. One of the particular ecoenzymes that UVM measured is a chitinase. Chitin, of course, being a component of insect exoskeletons, and regen produces a very significant amount of a chitinase enzyme. NAG is the acronym, and the presence of that ecoenzyme can serve the function of breaking down insect larvae, eggs, and kind of getting into their exoskeletons.
Okay, so there are some mechanisms that may help in reducing some of that risk. But it is something I think growers may want to be aware of. I love the idea of kind of what Victoria talked about, this idea of sort of recapturing and releasing some of these nutrients that may be tied up in the plant themselves and just less accessible due to the lignin and cellulose. But I do worry about heavy metals, pathogens, pests, and viroids now, especially with the hop latent viroid, as potential concerns. But over time, maybe we find with research that in a real world facility, it's less of a concern.
Yeah, those are all very real concerns to growers. So I hope we can start to address some of them more with the work that we do at Imio on regen.
Yeah, and I would add that, like, in an outdoor situation, in a greenhouse that maybe it has more exposure to outdoor air and pests and pathogens in general, I think I wouldn't associate the same level of risk. And in fact, I think I could see a lot of beneficial use cases in those scenarios. But I appreciate the dialogue around it, because I know it is. I think it's important to just have a conversation around it, and at least get people thinking about it. But that being said, Is there anything else you want to add about root or regen or the technology itself before I cut the show and we cut to Dr. Ben to talk a little bit more about the research at Gold Leaf?
I think that should about do it. Again, appreciate the opportunity to be on the podcast with you, Tad. It's a great, great platform that you built here. Great way to share a lot of wonderful knowledge with growers. We enjoy listening and appreciate the expertise that's shared on this, and we're really happy to be part of the conversation, and really excited to be working with you and the team at KISS, and to be offering our products on kissorganics.com very soon.
Yeah, thank you so much for giving us the opportunity to talk more about root and regen and how they came to be and what they could potentially do for growers. So really, really great to talk with you today.
That was Victoria Holden and Charles Smith, and you are listening to the Cannabis Cultivation and Science podcast. I'm your host, Tad Hussey. If you like the podcast, please leave us a rating and review and give us a follow on Instagram. We also have a Patreon where you can get insider access to the team at KISS Organics through Hangouts, as well as some cool perks and benefits for supporting the podcast. Lastly, you can sign up for our newsletter on our website homepage to stay up to date on the latest research and information. Thanks for listening. All right, we're back, this time with Dr. Ben Higgins, here to talk about some of the results using the Imio product Root over at Gold Leaf Gardens. Ben, can you talk to me about how you guys set up the experiment?
Yeah, so it was a pretty straightforward experiment. We used cloning domes, and we do have some other SOPs associated with our cloning normally, but because we were just really interested in how the Imio root performed against the kind of leading competitor, we held off on all those things. So this was basically just a plain water and the rooting hormone trial. And what we found was pretty encouraging. Survival rate was not significantly better, but that was great because we were still hovering around 80 to 85%. So we didn't see a decrease in using the Imio root, which is something we obviously were very cognizant of. And after that, the main thing we were really tracking that we were able to track was clone just growth. So we just looked at clone height. So all we did was basically the day that those clones went into the plugs, I just took a height measurement and I went from the base of the plant where it intersected the plug all the way up to that apical node. Over time, I've started to use that kind of as my height measurement because, fan leaves can go up and down and they're bigger or smaller depending on cultivar. So really just kind of keeping a standard reference point, that top apical node, standardized things pretty well. So I went from that, and then we just basically gave them a hand watering when they needed them, a misting, left them in a controlled environment, the dome, kind of standard protocol. And after 10 days, I popped the lid off and just wanted to get an idea of, if we were seeing any better performance and growth. And to set this up, we always try to conclude at least 2, if not more, cultivars. It's kind of, production dependent. So we had 2 cultivars. We had the first one was called Lemon Mimosa, and the second was called Hat Trick. And after 10 days, we actually did find significance in Lemon Mimosa. The average increase in height was about 2.8 centimeters, whereas for the synthetic rooting hormone, we only had an increase of about 0.73 centimeters. So that was a significant difference.
Now, Ben, just to confirm, what was the synthetic product that you were using?
We were using Clonex gel.
Okay. So this was root in comparison to Clonex. Great.
Mm-hmm. Yeah. And then, so the second cultivar Hat Trick, same thing, same protocol, same measuring techniques. And after 10 days, we did not see a significant difference between clones, between treatments. I'm sorry. the mean change for Imio root was 0.95 centimeters better, whereas that for Clonex was 0.92. So better, but not statistically better. The thing that we were also very excited about was we didn't see it any worse than Clonex, and that was kind of the benchmark for us to be willing to accept it into our program, which is as long as it held its own or did better, then we were happy with it. So that was just kind of the tip, though. After that, obviously I tried to get a couple of more. What's the best way to say this? A couple of more direct comparisons to see how Imio root was performing, and I was really focused on the roots. I tried weighing out the peat plugs prior, and then cut the clone off at the base, so I was just left with the root mass and the peat plug, obviously. But there was still so much variation in just how much moisture those peat plugs held onto that I didn't feel like I could get a very clear signal as to if the change in weight I was seeing was actually added root mass, or if it was just because that particular plug held on to more water. So I didn't go with that. But because of the serial sacrifice, meaning that I purposely killed clones to get at that question, I wasn't able to take as many replicates forward into production that I wanted to. So because we saw the really nice success rate and the promising growth, we then did it again on 2 other cultivars that are now, oh gosh, only about a week away from harvest. And so those went the full cycle. So then we'll get a really nice sense to see if we are getting any of these early onset kind of does grow, is the relationship between the plant and Imio, the increased in growth, does that translate to obviously the thing that most growers care about, which is, does it increase bud production, bud weight, dry weight, and all that fun stuff. So stay tuned for that. That should be coming within the next couple weeks.
Yeah. So the hypothesis then is that because Imio is the root product is a microbial product, we may get some microbial persistence that can last beyond just the propagation stage and
Right.
into veg and potentially into flower. So we can get to flower faster by shortening the veg stage possibly, and then also get increased yields overall is really the goal there.
There's a bunch of knock-on effects from that, right? So if you manage to get your plants, your clones to a certain threshold height, whatever facility or grower feels that, whatever that height may be, but if you can get there faster without obviously sacrificing anything else down the road, the big obviously business side of that is you can maybe squeeze in 1, maybe even 2 more harvests per year, because you are decreasing that veg time. So you could flip to flower quicker, you could harvest quicker, and then maybe, depending on your production schedule, get in a yield or a harvest or 2 more. So that in itself, obviously, if you were to crunch the numbers, would be a pretty impactful change in production. And then, yeah, exactly. From the microbial perspective, we want these plants to hit the ground running as fast as they can. And the lag between when you transplant from a somewhat sterile or synthetic environment into, what we do, which is the organic living soil, we don't know how fast it takes for those symbioses to develop. We don't know how long it takes for that bulk flow signaling between the plant root exudates and the microbes in the already present in the soil to, how long does it take for them to make that handshake? So if you can get that process started earlier on, I feel like you're helping out with not having such a lag time between that. And it could very well easily, this is all stuff that of course I'd love to be able to get at with a compound scope and actually see these things, but it's a tricky thing to do. But just to see the plants kind of take off a little bit more quickly, if you do manage to get more, instead of just that one big apical growth, if you get more branching and those things are, keeping up a little bit more, There's other kind of signals we're going to try to, see if this, relationship between early root colonizing by an organic microbial community pays off down the road versus just a synthetic molecule that kind of makes the plant do this rooting thing, and then it's kind of just left in a sterile environment to then go seek out those kind of organic relationships.
Now, Ben, did you measure the overall growth through veg to see if you did get increased growth in the Imio plants versus the
Yeah, we
I
did.
guess the Clonex ones?
Yeah, so we saw it for that first 10 days. Those were the statistically significant results that we saw for lemon mimosa. We didn't see it again in hat trick, but we continued to track the growth, and those that pace stayed constant. So in fact, for lemon mimosa, which was the one that we saw that big jump in height, Imio relative to Clonex, just by sheer randomness, the clones actually started for the Imio treatment. They started out a little bit shorter than the ones did for Clonex, but after 10 days, those totally switched. So you had, the Imio clones were bigger, and then we tracked that for another 10 to 12 days. Weekends kind of throw off production cycles a little bit for us, but right the next time we were able to transplant into our beds to flower, we took a point there and also looked at vegetative growth performance to make sure that pattern stayed constant, and it did. So it didn't seem like it was just this big bump and then kind of level off. It continued to rise, especially relative to the Clonex trial. That did hold true for hat trick as well, in that once they were on a growth trajectory, it didn't really change too much. So it kind of lends a little bit of credence to the whole, if you can get them jumped quickly, they kind of take off on that. Obviously, unless there's a hiccup in nutrients, or if they're running out of gas or anything like that. But if everything's there and they do have a good jump start, they tend to continue to stay on that trajectory. We don't see any fall off yet, and we certainly haven't seen any going into flower. I'm very anxious in the next week when we bring that room down. The plan there is just to take each plant individually, weigh it individually, tag them individually, and then that way we'll be able to know, here's the overall plant height at the end. Here's the wet weight for that plant, and then we'll track that all the way through to dry weight, and then we'll have a metric at the end, which is how much flower or jarrable product did this one plant give us relative to the other treatment that we did all the way back 3+ months ago.
Yeah, and I think the main goal of this is just, I mean, initially was just to have an organic option,
right,
that is consistent and can match, or really just match with what we're getting with Clonex and these other, synthetic products.
Yep.
So the fact that it seems to be doing so is, I would call it a win. And then in all this extra stuff with potentially better growth, potential disease protection, some of these things that they're starting to see, I think is really exciting.
Yeah, and as a scientist or nerd, however you want to slice it, it opens a bunch of really interesting questions, right? Because obviously, as a grower, you'd love it if hat trick had the same performance as lemon mimosa, which was, this thing just exploded, relative to Clonex, but it didn't. But I think the thing too, and maybe just kind of a more general thing for growers to remember is, just because you didn't get the result you were expecting doesn't mean you didn't get a result, right? So, now we have this kind of really interesting thing where it's like, well, lemon mimosa played very nicely with Imio, and hat trick not so much. So the question now is very exciting, which is, well, why is that, right? So then it challenges you and it focuses you to maybe think about other aspects of your grow, where one was one bed mixed differently, or what can I control to maybe help explain why we saw this result. As long as you're critically thinking about unexpected results, there's a lot of potential for growth there, no pun intended. So it's just something for, I think, growers to keep in mind, which is, if things don't go, if your hypothesis is not supported fully, that's still a result that I think is merited at least a couple of thoughts and a conversation as to why. And I know in a production facility, getting at the why and the how tends to not be the most important pathway, because it doesn't really necessarily lead to additional revenue, right? And this is all obviously a business that we're going for here. But on occasionally, the why can be more universal and then applied to other things that might be limiting growth, right? So just keep things flowing, critically thinking. It's a really, really important tool, and a lot of benefits can come from it.
Yeah, and the last thing that I want to mention before we sign off is that not every product or method or technique that we try out gives us a successful result. We've tried a lot of products over the years through Gold Leaf, and many of them performed below what the control was, which is essentially what Gold Leaf was already doing. So I think we've come a long way. And your initial podcast I did with you a while back showed just how much you were able to make improvements just by running some of these trials.
Yeah.
But I do want to highlight, like, we've tried other microbial products in the past. We tried this one really cool sounding Archaea product where the trial just kind of fell apart. We tried other things on the microbial side where we just didn't get a noticeable result. So yeah, I think it's an important thing.
Yeah, it's a really great point. And the way I kind of couch all of that, like, well, this worked, this didn't, but maybe it did for another facility, right? I mean, the thing to remember here is that, we focus very heavily on just cannabis because that's obviously what we sell. That's what people take pictures of. That's, the main species we're targeting in all of this. But when we're talking about living soil and microbial communities, I mean, Tad, do you want to take a guess at how many different microbial populations are in a given bed? Because I don't.
Yeah, we know it's large.
It's a lot. So you're managing quite a lot of relationships, and these relationships are evolving over time, right? So you put Imio root in at the beginning of a cloning round, it's going to interact with a host of other different populations, all given a specific environment. And I mean, one of the best ways that I've always liked to explain this is, the two famous tennis players, right? Roger Federer and Rafael Nadal. They're legendary. And one of them, Nadal, was an absolute monster on clay, where Federer was just almost unbeatable on grass. And then when these two played each other, their record was almost purely determined, with a couple of exceptions, but almost purely determined by the court they were playing on. So if they were on clay, Nadal would win. If they were on grass, Federer would win. In fact, they set this thing up because they wanted to take the environmental perspective out of it, and they made a tennis court that was half clay and half grass. So they put Federer on the grass and Nadal on clay. And so it's always a nice reminder to me that when we set up these experiments and these trials, that as much as we'd love to say this holds true for everything, and sometimes you get lucky and that can happen, it's rare, but it can. The species that are interacting in your soil with the plants are all taking place in this backdrop of the environment that they're in. So you could have the exact same genetics, the exact same microbial populations, but just put them in a different environment and those relationships will change. Some antagonistic ones might become symbiotic or vice versa. So because stuff isn't working, kind of like you were speaking to, we've tried things that haven't worked. We tried things that haven't worked. You keep trying things. And as long as you can set aside a couple of beds or pots to just let those little things, that little subset of your grow answer the questions that intrigue you, there's a lot of power to that. And, just going back to that tennis example, like the environment that they're in really does set the stage for how these things interact. So just because a microbial product maybe didn't work, it could very well be that maybe it didn't have anything to do with the microbial product at all. It was just that the VPD was off in the room, right? Or it was overwatering, or, so that's why replication and trying more than one cultivar, these kinds of things are really important. However, they obviously take a lot more time. So there's a trade-off, but it's always exciting.
Yeah, and on the flip side, I think there's a lot of microbial products that frankly just aren't that great.
Absolutely.
And that's part of the reason it took us over a year to, make this podcast to move forward with Imio Root, was because we wanted to make sure it was something that we had some data on ourselves, in addition to the research that they shared, to know that it's going to be something that we feel good about sharing and promoting to the community at large. So,
right. And for those that are very much into, organic processes from start to finish, as we are, this seems to be a really great alternative to some of the more, common synthetic products that are out there. At least give it a trial and see, if it does work in your facility, and use your own results to inform if you want to scale it up or make any other decisions.
Cool. Well, thanks for coming on today.
Anytime, Tad. Thanks so much.